It is very much in the spirit of thinking where we're going, moving forward, and particularly thinking about translational science and what that actually means, that this symposium has been organized. And it gives me great personal pleasure to introduce our keynote speaker. Ruth O'Hara is the director of Spectrum, which is the Stanford Center for Clinical and Translational Research and Education. She's also the Senior Associate Dean for Research at the Stanford School of Medicine, and the Lowell W. and Josephine Q. Berry Professor of Psychiatry and Behavioral Sciences at Stanford. Ruth has a long and distinguished record in psychiatric research, but she's also really a wonderful leader and thought leader in terms of what is important in clinical and translational sciences. She's going to talk to us today with a broad vision of what translational science really means and how we're going to move it forward. On a personal note, it is a great pleasure for me to introduce someone whose accent I can completely understand.
Thank you so much. Such an honor to be invited here today. I have to say that I have many ties in multiple ways to Washington University and to the School of Medicine. Your very own Eric Lenz, chair of the Department of Psychiatry, is someone that I grew up with academically through the years. Both of us were members of the Career Development Institute, an NIH-funded institute to help grow the physician scientist and the PhD scientist through their early career development. I'm also very fortunate to have actually family in the audience. One of your own health economists, in fact, a preeminent health economist nationally and internationally, Dr. Timmy McBride, happens to be also a brother-in-law. So thank you, Timmy, for giving me my coffee this morning here and helping me out. And in addition, I have to say that we become colleagues and family as part of the broader CTSA community. It is really quite something, and I'm only a relatively recent member to that community, but it brings such impact and it's such an honor. And I want to speak a little bit about that today and the changes that we've seen. I would say, to echo the comments made by Dr. Lowe earlier, is that it is a very challenging time. And just as during COVID, we saw the CTSAs step to the fore and be able to provide so much support, they were so well positioned to pivot and to support. I think we're going to see that now. I think indeed we are seeing that already. So with that, oh, and just one last thing. I do want to emphasize that it's actually not an eligibility criteria to be a CTSA director to be from Ireland, but it is actually striking that I believe 5 of us currently have that particular background. So just to give a broad overview here, and I go next. So, yeah, the middle one. Thank you. The NIH Clinical and Translational Science Award Program is a very, very impactful one. It's run by the National Center for Advancing Translational Sciences, NCATS, and it has approximately, I think it's between 62 and 66 centers across the nation at any one time. The director is Joni Rutter, and we have a wonderful leader who actually oversees the CTSAs, of which your Institute for Clinical and Translational Sciences is, of course, one, and so ably led by my countrymen, but Dr. Michael Kurilla oversees the full component. And the aim of the CTSA is to develop innovative solutions to improve the efficiency, quality, and impact of bringing observations in the laboratory, clinic, and community into interventions to improve the health of all. And in fact, this last line here has been, for the last few years, the true mantra spoken of at every strategic planning meeting, in every meeting where you'd hear Dr. Rutter speak or Dr. Kurilla: more treatments, more people, more rapidly. I'm really bad at this here. Oh, thank you. You can tell I haven't had enough coffee quite yet. So each CTSA itself is a program that encompasses research, innovation, and training in support of the clinical and translational research enterprise. And the program actually occurred in response to the fact that up until the 2000s, really academic medicine was less emphasizing clinical trial research and infrastructure, and even translational medicine, but really the emphasis was on the basic science. And to be very honest, that's been a very, very effective place for academic medicine to reside, because it has produced so many treatments, so many innovations. We look at what's happened in the last few years in terms of cancer therapies and the CAR T cells, innovations in AI being applied now to real-world data. I mean, it's a very exciting time, despite recent challenges in the field of biomedical research. So we are very lucky to be one of over 60 CTSAs that collaborate to share innovations in training, research, and research tools and procedures. And of course, with your own CTSA here, you see this on a daily basis. And the overview that Bill gave was superb in terms of underscoring the kind of things that are supported by CTSAs that traditionally might otherwise not get that kind of level of support. So at Stanford, the primary functions of our CTSA over the years has been to provide that infrastructure. We're an institution that traditionally has favored basic sciences, perhaps at the expense of our clinical trial and clinical infrastructure portfolio. So we now have clinical research units within each department that are actually supported by our CTSA. And they come together on a regular basis to learn the best and most effective practices for onboarding trials. We're still learning a lot in that domain, let me tell you. Also, we have a wonderful ambulatory center for the conduct of ambulatory trials, including our cancer trials. We have a biobank for collecting, storing, and assay of biological samples, which really has been a huge way that we've been able to bring actually different studies together. We have a vast amount of regulatory support for our faculty. We also have an honest broker system for participant engagement and participation in our clinical trials. So rather than actually having some of the conflicts that can exist when an individual investigator is trying to actually bring a patient to the table, we actually have a system that does that for them, that actually makes sure the word is being distributed about the study itself, and then to help with making sure that any patient that's coming to participate, their eligibility is reviewed. We have an opt-out system where actually patients coming into Stanford can elect to be included in a large registry that we can reach into through this honest broker system, and we wouldn't have that without CTSA. And we're also part of the Trial Innovation Network, which is a CTSA nationally supported entity, so that any individual investigator, particularly in the domain area of things like rare disorders, can reach out to other CTSAs to see if they want to participate in that trial. CTSA supports a tremendous amount of innovation in data design and analysis, and that's a strength locally at Stanford for us, so we capitalize on that. And then last but by no means least, in fact, the primary aim of our CTSA is to train the next generation of clinical and translational researchers. Our K-12 scholars, our pilot study program is very much in support of our junior faculty and our junior investigators. We haven't yet got our T32, so we may model that after bills. And we have over 20 educational offerings that support training people who are actually already quite versed on the next level of clinical trial implementation to those where, in fact, it's their first time learning about clinical trial infrastructure and support. And we have some very nice locations at Stanford to conduct this work. And this is our website, which is really the home to our CTSA, where people can go and are navigated and instructed towards to learn more about the resources. I will say, and I will say that this is perhaps using a journalistic term a little off the record, but we have morphed our website over the last few months to really provide as much information to our faculty, to our trainees, to our staff regarding the current situation. I think one of the things that a CTSA teaches its investigators is it's really important to be aware of multiple domains that actually impact your research. Oftentimes, as scientists, we're really drawn to the specific hypothesis and area of inquiry, but we've learned, if we didn't know before, how important it is to be able to follow legal challenges and to follow actually cases that are coming before courts and to understand and interpret executive orders. I mean, this is perhaps not something that we all think about as a skill set that we might need. But I think going forward, it's very clear that these skills are really important as each of us carves and pursues our career trajectory. So we actually want the big link on the CTSA now is to those cases, because oftentimes when you're sitting waiting to hear, you know, what's the next thing the institution is doing, it can be very reassuring to be aware of the national cases where, in fact, a temporary restraining order may be put in place or a preliminary injunction. Or it might also give you some understanding, perhaps, which can be very frustrating, why you haven't heard from your leadership. It's quite a balancing act at the moment to actually engage and respond to all of the various components, but to do so in a judicious and careful manner. So sometimes it can take a week or two to actually get information back to our communities. And I would say this is a time where it's really important to be patient. We also set up a portal, and this is behind us, they would say our firewall, where investigators can actually ask questions, individual questions, questions specific to their own situation, in order to really get them addressed in a way that we may not be able to publicly address everything at the moment. So that has been a very helpful aspect of our CTSA, is that we really sit in a place that has that role at these kinds of times. There's several unique aspects to CTSAs that I think are not always advertised or communicated effectively, and particularly speak to some of the aspirations of NCATS and their strategic plan. CTSAs are disorder agnostic. They don't actually follow, like the Cancer Institute's, a specific disorder. So there are lots of collaborative opportunities. And I think the vision now of NCATS is to actually try and accelerate the production of things like the toolkit that you saw on the revolving slides here, that you have developed here at WashU and that is disseminated now throughout the full CTSA community. It is really an aspirational goal of NCATS to be able to develop and disseminate throughout all of the CTSAs and the communities beyond the best and most effective procedures for making that translational research jump to implementation. CTSAs, as a result, have evolved from a resource emphasis to emphasizing the processes and approaches integral to the science of translation. They also have, as an institute, a significant emphasis on real-world data, digital health, repositories, and artificial intelligence. They really would like to be the place where a lot of that particular area of science is developed, encouraged, and supported. And it's very clear that this is an area that is important to the new NIH director. There's a significant emphasis on communication across CTSAs that I think is particularly valuable and that we could model potentially in other domains that actually accelerates translation, and I'll speak to that in a little bit. So over the years, we've had thousands of publications at Stanford that have been supported and availed of some of the resources of CTSAs. This is just a small example to capture the heterogeneity and also to capture the kinds of domains that we focus on, like digital health and so forth. But while we've been very effective this way, and we can always show that we're a good investment for a federal dollar, at the same time, strengths can be our weaknesses. So multiple basic clinical and translational studies and publications are supported. Hundreds of trainees have been mentored into academic and industry and beyond positions. But yet, the valley of death, that transition from actually the first clinical trials, or even the second and third, your phase 3 and your phase 4, into really scalable interventions at a community-based level has been the challenge. And there's a lack of sufficient translational practice or principles that we have developed over the years. And that's become really the focus of NCAT, is to provide more systematic guidance for spanning the full translational research chain and making sure that we don't drop off at the phase 3, or indeed even in the initial periods of implementing in the community. That's where sometimes we don't actually manage to sustain the impact. So as a result of this, one of the great leaders in NCATS and the CTSAs, Chris Austin, who was a major figure for years leading that entity, said, "No more free stuff." Now, that doesn't quite mean that CTSAs are abandoning the phenomenal resources that have really meant that we've increased dramatically our clinical footprint and clinical trial footprint within academic organizations, but they are thinking differently about it. So the main area that they have focused on in recent years is really putting a stronger emphasis on what Bill talked about as translational science. So what does that actually mean? Well, when they started, just to give you a little history here and context, CTSAs were actually housed within, or at least the initial proposal for CTSAs were overseen by NIH's Center, not an institute, for Research and Resources. So it seems very appropriate that, in fact, CTSAs would have a strong resource focus. And through the years, they started with an initial planning grant. We got our first CTSA in 2008. And they've morphed to this, let's now really establish and abide by principles of translational science so that others independent of the domain area can pick up the baton and be able to actually more effectively transition from the early stage trials right through scalable implementation. One thing that they made as a change that I think is very helpful in this regard, and it's oftentimes that the devil is in the details, very practically, we actually are now a grant that's 7 years. So when you renew under this new notice of award that came out, or funding opportunity that came out in 2022, you actually can have a CTSA for a 7-year span to give you time to achieve actually that implementation piece. So that's been a huge addition. Despite that, when the NOFA came out in 19, that's my age telling me there, 2022, we actually had a lot of disagreement among PIs of CTSAs about what it actually means to do translational science as opposed to translational research. And in fact, the NOFA was modified in response to that. And part of it, I think, is the fact that the field of science of translation is evolving. Translational research, if you're to give it a very simple definition, is the process of conducting investigations along the continuum to bridge discoveries from bench to bedside and back, but actually then implementing in actually the full community-based setting any kind of treatment is much more challenging. So the science of translation is a field of investigation to understand the underlying principles of each step of the translational process that can be applied broadly to facilitate and accelerate translational research in all domains. And I think what's a little problematic, and has been for CTSA PIs, is those are not shovel-ready science of translation principles. They don't actually exist in any cohesive way. And in fact, I think that's the goal of NCATS, is to produce a way of thinking about the science of translation and the principles inherent to that that might actually achieve the dream or vision of the science of translation. And many people have said, isn't this just the same as implementation science? Well, CTSAs would argue no. It is, in fact, really important to be thinking about that translational component as early as the very basic fundamental discoveries that lead ultimately to the treatment you want to implement. And that's a very interesting perspective, but it doesn't mean that those of us who are actually trying to implement a science of translation program can have actually ready procedures or principles to call upon. So how do we manage that? Well, at Stanford, what we did is we said, well, what entities do we already have ongoing that we would think would be particularly valuable for the science of translation? And we developed what we thought were the optimal pillars of science of translation within our program. And I think in the initial periods, before we gain from each other iteratively, this may be the best approach. So we have a strong program on rigor and reproducibility. And basically, non-rigorous or non-reproducible science does not translate. It's been a fundamental barrier. So because we already had an existing program that could address that, we thought we'll make this one of our pillars of the science of translation and focus on developing principles out of that component, and that we would also take those already to our trainees in all of the programs, from undergraduate right up to our postdoctoral and indeed junior faculty. We have a very strong emphasis on team science. A pass-the-baton approach we've long known doesn't work. Interdisciplinary is key, but it's easy to speak to, it's harder to actually achieve. And I'm going to talk a little bit about all of these in a moment and how we're tackling them within our programs to actually implement these as sort of standard entities and components that you need to think about when you are actually an investigator within our CTSA, and then to spread that more broadly. Community-based research. You can see the language change already. We now talk about community-based research and patient participation. It is extremely important when we are receiving guide memos about civil rights to underscore that in the domain areas of clinical trials, the goal is to actually bring as many participants as possible, not to exclude participation. We may have eligibility criteria that actually are specific to the disorder or focus on that disorder that we are considering, but we are not in the business that I've ever seen of actually turning away individuals unless it was for the eligibility criteria that have been set up ahead. We want all comers, and we have a lot of systems within CTSAs to encourage patient participation. And it's essential for generalizability. I mean, generalizability is key to good implementation and bringing clinical trial outcomes to our communities. It's critical. So that's the place where we're emphasizing that this is a critical component, and we don't use the word inclusiveness anymore, but it is all patients having the ability to participate. The other place that we think we have a lot of strength in and that we could do more is in the domain of real-world data, large databases, biostatistics, informatics, and data science. In fact, an increased recognition at the CTSAs about how epidemiologists, biostatisticians, AI experts, data science experts, people dealing with electronic health records and informatics, that they're all tackling the same problems. They created an entity called BIDS, which is essentially that biostatistics, informatics, and data science component to embrace all of that. They become very important platforms, not only for data hypothesis generation, but also for validation and testing, and can be very, very important for early stage career in a way that so many of our early stage investigators are struggling with these very small clinical trials for which they have received perhaps a small grant or a small foundation grant or a little bit of an IK award, and they start with that, which gives them great experience, but doesn't give them always that important paper, doesn't always actually answer a question that might have the answer to some extent residing in some of these databases. And then we also have a strong health policy component because we recognize that's essential for implementation and scaling. So just to speak a little bit about the reproducibility and replicability problem that actually the National Academy of Sciences has even issued reports on, I will confess that we had to take this very seriously locally because we had a precedent where there was considered to be a lack of sufficient attention to certain figures in their papers that seemed duplicative. And even though some of these were quite a while ago, and even though we acknowledge fully that our president, Mark Tessier-Levine, was cleared of any type of misconduct, it underscored for us at Stanford as a community, we better do better in this space. And to that end, we have a program called SPORE, which is the Stanford Program on Rigor and Reproducibility. What makes a study rigorous? Well, it really refers to some of the simple things that we have all learned in our fundamental biostatistical courses, but that, what I call statistical hygiene, but that absolutely we can forget and very easily forget as our careers become busier, more complex. It refers to a well-designed plan and adherence to methodological best practices according to the National Academy's report. It is the soundness and precision of a study in terms of the design, execution, data collection, and analysis. And it is something that we say, oh, don't we all know that? But actually, it's amazing how quickly we can abandon some of the most fundamental principles, like, fudging your eligibility criteria on occasion, or perhaps not thinking about why you're excluding an outlier and rationalizing that sufficiently. We are going to be called upon more and more to actually speak to rigor and reproducibility, and it's going to be a critical domain for us going forward, and I think CTSAs can be real leaders. So some of the components that we particularly imbue into our courses are, what's the quality of the research question? Sloppy research questions can result in really non-rigorous science. The fit between the question and the design, the quality of the design, the quality of the conduct of the study, the analysis interpretation, and oftentimes, particularly at early stages of career, you don't always recognize that you need actually support at every one of these steps. Most of us coming out of the various programs might think, well, we can manage the design. And that's where some of the informatics and biostatistic units that are integral to most of our CTSAs really can play a very, very strong role. And so we've incorporated a lot of this into our training programs at all levels because it's critical. But it's also critical for senior investigators. And this is, raises some fundamental questions. Should, in fact, very senior administrators with huge roles still sustain leadership roles in their labs? There's some existential inquiry that we have to do here. Institutions need to do their part. They need to acknowledge that, in fact, if you're going to have very rigorous and reproducible findings, it may take longer to make sure that work has been done effectively. So modification of the expectation at an institutional level of how many publications a junior investigator might have is some of the practical considerations we have to engage in. So it's easy to talk about research rigor, but it needs to be adopted not just as another course in our Essentials of Clinical Research program, but also institutionally to recognize what that means and the impact it may have on actually early career, on even more advanced stages. There are more sort of institutional questions to be addressed here. One of the things that we hosted a symposium on last year was the problem that arises with rigor and reproducibility when you don't publish negative findings. And so we were very lucky to have Holden Thorpe from the editor of Science attend and say, there is a tremendous pressure on junior investigators at all stages to have that exciting finding. It's not as exciting to turn up and say, I tested this, I didn't find anything, then we, tried it out in one of these larger databases and it doesn't seem to be an important relationship there. We have to change how institutions judge those papers. And as a result of that symposium, Holden actually has said he will put aside some part of Science's portfolio every so often to make sure that they're actually reporting on some of these negative findings. And that's the kind of systemic change that we really need. Similarly, team science. If there's multiple parties speaking to each other at the same time, and we put a lot of effort in bringing those groups together, how do you allocate credit academically fairly? And this is another place that institutions have to step up and recognize that a project you're engaged in that is team science-based actually gets the acknowledgement for your place as maybe second to last author or second or third to first. And that's annotated in a way in your CV that acknowledges your contribution. I think we have to come up with better ways to make that acknowledgement. We have significant expertise at Stanford in AI. It's the particularly exciting domain of the moment, but it needs to be accompanied by expertise in real-world data, bringing various data repositories together. And I'm just going to give one example here because I think it's helpful. And how am I doing time-wise, Bill? I'm close to the end. But one of the things that I would say is I had a junior investigator who got a foundation award, about $20,000, to actually conduct a clinical, we wouldn't even call it a clinical trial, but a two-group comparison on the relationship of suicidality to sleep. Small amount of dollar could fund 10 in each cell. What are you going to be able to tell from 10 in each cell? It's too little for good pilot data even. It's very important for learning how to run a study, and we accepted that foundation award with great excitement. But this is a very statistically savvy junior investigator, and I said, well, have you looked at any large databases where in parallel you could see if you could address this question? No. And this is someone who's well used to large databases. Why didn't you think of doing that? Well, never crossed my mind. Okay, is there any out there? Well, actually, now that you mention it, there's this one with 10,000 individuals that has some proxy measures for what we're interested in that could be a very good place to look at first. So how do you find it? How do you go about, maybe actually stitching it together with other databases? So one of the things that we did in our renewal for the CTSA, we're renewed now till 2031, we actually said we're going to teach a course and for our trainees on how to think about actually even the use of these large databases, much less how to do it. Because I think in many cases, we now need to start paralleling the use of these large databases for the questions in hand, while simultaneously doing the more on-the-ground operational clinical trial or two-group comparison, as it usually is at that stage. There's so much you can learn, and it also makes for a much more effective early paper for that investigator. Can also change the direction of how they're thinking. So that was one of the promises we made, and we will keep. We're working on that already. And talking about reproducibility, we had a paper come out the year before last on epidemiological approaches and how actually difficult it is to reproduce many of the studies that exist more broadly. And part of the reason is even very subtle changes in outcome measures. I mean, myself and Eric Lenz for years have worked on depression. There's so many different measures of depression, different components. So it often looks like we're saying, well, this isn't reproducible. But actually, in very few cases do we have the identical study with the identical measures to actually compare. And one place where that may occur is these large databases, because you can put aside a certain amount of randomly selected individuals. If you have a sample of 10,000, you can call out, 2,000 to 3,000 to actually then, once you have your findings, see does it replicate in that additional sample. And it's an opportunity for us to do more rigorous research. And the last thing I want to talk about is the scaling of this. So one of the things when we do this course and we get it developed is to actually disseminate this particular course on how to access, know when to, and go to and use these large databases, or even in some cases lakes. One of the things that's particularly unusual about the CTSA is they have a phenomenal method of communication. So they have a steering committee. I was very fortunate to co-chair this with Mike Carilla last year, that 10 PIs are selected to serve on for a few years. And then they rotate off and another PI comes on board. But what that CTSA PI does, they're obliged to meet monthly with 6 other PIs, so that every piece of information that comes through the steering committee that meets twice a month gets disseminated rapidly, including updates on the kinds of programs, like, for example, this course or a trial innovation network, or some novel therapy or approach comes to the steering committee and then gets fed back to the community. And not a lot of institutes have this, hub and spoke model, but I think it could be an invaluable way for NIH to start thinking, and for us to start thinking of ways that we can get the information to others when we actually have a committee. And we're doing this at Stanford by leveraging our centers more than individuals. That's a shift in our CTSA model, and then working with other networks. So you take a network like CTSA has developed and get together with the Gregor Network on Rare Disorders, and you can produce some tremendous collaboration and impact. So on that note, I'll just summarize that our vision continues to expand our clinical and translational research enterprise, but we think it's particularly important to increase our efforts in team science and rigor and reproducibility. We want to expand research participation and the research teams necessary to optimize that. We also want to help define with NCATS what it really means to have principles of science of translation, to train the next generation, and to utilize more and more, when we can and where appropriate, real-world data electronic health records, and artificial intelligence, bioinformatics, and fundamental biostatistics to conduct that science of translation. Thank you so much for your attention. And I have a tremendous picture of everybody on our CTSA, but I couldn't get it to send this morning over to Chris, so my apologies, but I assure you this is supported by a huge team just like Bill has here.
Thank you so much for that. That was a wonderful overview. I have a question about rigor and reproducibility with data repositories. I wondered if you have guidance for us to model on how you set safeguards in place for appropriate use of public data versus making it available without any restrictions.
Well, super. You know, you've captured several really important things. I think there are some really strong efforts to take large public databases, for example, with Medicare, Medicaid, and other large healthcare databases. The problem is the harmonization. It really can be a significant challenge. And it needs resources, so we dedicate in our CTSA significant resources to actually trying to do that stitching, for want of a better phrase. It may be that we never reach a point that you know, we don't have categorical types of data. I think one thing that we could do, and we're trying to do within this course, is really advise individuals how to set up their databases in a way that makes for much greater ease of, in fact, that synergy. I'm sure many of you have been involved in efforts to, you know, migrate REDCap data over to EPIC and from EPIC to EPIC Research. We didn't do a good job of being proactive enough. I think when we were setting up those systems and thinking down the line, we couldn't possibly see, I think, the future, but I think we have to try. So a big part of this course will be actually trying to get individuals to think ahead of time, and for us to actually really develop what's the best guidance rather than it being individual to every different system. The privacy issue is really critical, and we've seen that in many cases where even when a very good database has been established, like the UK Biobank, that they still ended up with problems because they hadn't engaged the community of certain disorders. So for example, Simon Baron-Cohen, who does autism research, ran into difficulties when they wanted to actually disseminate that data broadly, including the biologic data and the genetics, that they got pushback when the project was almost completed on their end because they hadn't engaged at the earliest stages the community voice that would have told them that individuals with autism spectrum disorder have a lot of concerns about privacy and particularly chain of custody. So thinking these things through, I think, is very important, and I think we have to do the privacy in parallel, and we have to make sure that community engaged participation occurs at the earliest stages, even if it's very fundamental basic research, because it can come back to haunt one later down the line.
Hey, thanks for the shout outs, Ruth, but I have a hard question, I guess, which is about what do you think is the role for the science of science in improving measurement in translational science? I'll add to this a long comment. I've noticed that you know, the truism that methodologic advances precede scientific advances. And when I look at fields like genetics, molecular biology, and basic neuroscience, and now translational neuroscience, neuroimaging, there's a relentless focus on improving precision of measurement. In my own field, mental health, clinical trials, there's very little, if any, focus on that. And as a result, one might ask? Should we be focusing more on that?
You know, it's a great question. I think it's complex. On one hand, one would say that measurement has to be focused on. I think one of the problems in some domain areas like behavioral health is that we have complex disorders. And so actually saying that we have any single measure of depression that is going to be the ultimate outcome measure is really challenging. And I would say actually, Eric, that I think that a limitation on our constructs of rigor and reproducibility reflects the expectation that we can do that. Because I don't actually think we probably can, and if we were to, I mean, we've come up with some measures, you know, PHQ will measure, you know, and it's such a single, measurement that leaves out so many symptoms and phenotypes, and probably has impacted the research on genetics, because we know that as we've started to parse apart different types of depression, we see the markers now that we didn't see 10 years ago in the domain of depression. But I think it's really time-consuming and challenging, and it may not give us the yield immediately that our biological measures have given us. And even there, it's taken a long time. I mean, we're now at the stage where we probably have 5-6 neurocircuits that we say, okay, we know that when that circuit is activated, we're doing X. That reductionism, in a way, can be a challenge. And I think it gives a sense that we're less rigorous. If you were really to reproduce with the exact same measures some of the behavioral studies that have been particularly vulnerable to criticisms of non-reproducibility, I think we'd find they're non-reproducible because they're not measuring exactly the same thing. So I think you're absolutely right. It's a critical issue, but whether our approach should be arriving at an optimal outcome measure in ways we've been able to do with biological data a bit better, I'm not sure that's the approach.
Additional questions, thoughts?
I think you've one there. Yeah, this here to the left.
That was a great talk. Thank you so much, Ruth. I have a question. Could you comment on what is the approach that the CTSAs nationwide are having to be able to embrace small programs into moving the science forward? I mean, most of these hubs in the CTSA have great infrastructure, have great resources, but there's great talent outside of these big hubs. And I know that sometimes we have small affiliated institutions, but it's still hard to bring them in and be able to move the science forward. Could you comment what's going on nationwide to be able to spread that love?
Yeah, interestingly enough, under the last NIH director, there really was a mandate that we were insufficiently getting out into the rural areas. That was a real gap. And there are CTSAs that are embracing that, but perhaps you always, put your best foot forward, particularly when you're going to be going in for a renewal and you want to bring the programs that are particularly effective locally. But, Bill showed a great slide about the importance of interacting even just locally here in a really well-resourced urban area with your community-based partners and hospitals. I think CTSAs are going to have to do more of that, and we're going to have to do more out-of-the-box thinking about actually getting into areas like primary care setting and making sure that our selection of PIs that serve on the steering committee reflect those more those smaller CTSAs maybe that are a bit distal from the urban hubs. So those could be practical ways, but it's such an important point.
So let me ask one final question. Well, I actually have lots of questions, but I'll ask one. When you talked about translational science and the need to bring it really early into the at the very beginning. We've had discussions like that here because we were one of the earliest CTSA hubs to start implementation science. And Noel Proctor's in the audience really led that for many years. And we really have thought about it as an expansion of implementation science rather than a different component. The challenge is, how do you make this, and I don't know if we certainly don't have the answer, but I'm curious how you would respond. How do you make this into something that people who have at the discovery side, they think they have a target, they think they have a new drug. How do you make them realize that this is really important if they're ultimately going to get benefit?
Well, it's a great question. How we're doing it locally is we're making it an integral part of all our CTSA supported educational programs. So, we have, very similar to yourself, we have an intense clinical trial and research study program called ICER, which is a year-long program for junior investigators, senior postdocs, instructors, and junior faculty. And it's an integral part of that, as are all those pillars. And then for our K-1-2 scholars, we're doing exactly the same. They have to have a mentor who's actually at the community engagement and participation interface, who mentors them on why it's so important to have this knowledge right now, with examples of where things have gone south, where things can be improved, and where essentially your very innovative ideas not making that step forward because you haven't avoided that particular hurdle or barrier. That's how we're doing it now. Nancy raised a great point yesterday that I actually fully agree with, in terms of this interface or even parallel or substitution for implementation. That's not at all, I think, what can happen. I mean, I think exactly you're right. It's an expansion and part of actually a collaboration at the implementation stage. And we're very fortunate to have an NIH-supported implementation center at Stanford as well that we work with closely. But Nancy said, are we going to get, science and translation investigators, and will they be subsequently funded by NIH? I'm not sure that we can. First of all, we're still defining what it actually means to do, science of translation. What are the principles? There really has to be far more deliberation about that, and some of it will be informed by saying, okay, well, here's these 4 programs where we look like we're doing something akin to science of translation. And maybe if someone's going in with an Aura 1, they really go in for one of those areas with a focus that augments science translation, but is not coming across as that's the primary focus. And I have to say, I think that's where we are, and it's going to be iterative.
With that, I want to thank Ruth again for a wonderful exposition on where we are and where we hope we're going with clinical and translational science.